Thank you very much, Lexley, for the introduction. And also thank you everyone for attending today's webinar. So what I'd like to talk to you today about is gene editing technologies and their application in biomedical research, as well as in cell gene therapy development. So I'm hoping to cover the following contents. I will give an overview on gene editing technologies, including the most advanced gene editing technology on the market. And then I will discuss application, especially research versus clinical application. And then I will focus on talking about gene editing technologies in iPSC. And then I'll touch on some of the other applications using gene editing for antibody discovery, screening, and of course for in vivo animal model generation. And then at the end of the webinar, we have time for question and answers. So what is gene editing? As most of you probably know, this is a really revolutionary technology, especially CRISPR. Basically, gene editing allows precise modification of the human genome. And so the process usually involves specialized enzymes and also involves guided RNA. So in this case, for Cas9, for example, is guided RNA to target specific location in the genome and then make desirable genetic modification. And so the tools I will cover today will include nucleus, which where CRISPR-Cas9 it belongs to. And then also mention about recombinases, particularly serine integrase. So that's what TARGATT technology uses. And then I will also discuss fusion proteins that performs gene editing. So here we listed several challenges in gene editing, so which we'll talk about throughout the webinar. So one of the biggest challenges is really off-target effects, because that really imposes safety concerns. And then, of course, editing efficiency is very important. That really confers efficacy to a drug product. And genomic integration, stability of genomic integration and where it is integrated is very important. That also is a concern of safety. And then another thing is payload limit. So I will mention about some of the technologies, especially the current state of CRISPR-Cas9 limits the size of the gene you can insert it, so which could be a problem for clinical application. And then immunogenicity is a safety concern too, because some of the gene editing components could cause immunogenicity from the host. And then another important thing is really freedom to operate, especially when it comes to therapeutic applications. So I will also discuss a little bit about that. Oops. So CRISPR, as most of you probably very familiar with, is like I said, revolutionary technology. And Jennifer Doudna and Charpentier, they received Nobel Prize for discovery of CRISPR-Cas9. And so basically, this is a nucleus. Cas9 is endonuclease. It makes double-stranded DNA breaks. And so that's what CRISPR does. And once the double-stranded DNA breaks are made, then the host recombination system kicks in. And so in the presence of donor DNA, either in a single-stranded or double-stranded form, the double-stranded breaks get repaired so that at the same time, the donor DNA get inserted. So that really results in insertion of the donor DNA. And if there's no donor DNA present, and this double-stranded DNA break will cause indel, that is deletion or insertion, and then basically, in most cases, will cause knockout, a change, a frame shift of a gene. So that's how CRISPR works. And however, in the host cells, this pathway, which is called non-homologous end joining, is the major pathway. So that's why knockout is very efficient using CRISPR. And then HDR, homologous recombination, is very, very rare event. It's 1 in a million. So that's why insertion using CRISPR is not efficient at all. And so there other CRISPR-related technology was discovered. So one of which is base editing. So base editing is basically a fusion enzyme. So it uses the CRISPR component, the Cas9 nickase, and then fused to a deaminase. And the traditional base editing has the deaminase fused to the end of Cas9 protein. So this is important because I will talk about ASC's active base editing, which has different conformation here. And and this deaminase basically changes nucleotide from C to T, for example, right? So that's how base editing works. And base editing works in terms of for off-target event, and it's better because it uses nickase. So it only makes a nick, not double-stranded DNA break. So that helps with reducing off-target events. But to some extent, it still happens. And so we have a technology which is our partner, Base Therapeutics, they have developed, is a base editing technology, but which with much higher accuracy. We have not detected off-target events using this technology. So the difference is really at the non-editing stage, the deaminase component is really it's buried in the protein, so it's not exposed to target DNA. So there's no event, there's no cutting, right? And then when the guide RNA leads the enzyme to the target DNA, and there's alteration in the structure of the protein. At that time, the deaminase component is actually exposed, and then results in the change of base from C to T, for example. So that's really provide almost the highest safety profile in terms of CRISPR technology. So this is just some data to show that. The main thing I want to talk about is really off target. So for the same target region, and in HEC293 cells, we detected close to 0 off target, whereas using beams, base editing, we detect 725 off targets. So this really shows the high safety profile that AccuBase can offer. And so this just kind of summarize the extension of CRISPR technology. Right? So originally CRISPR-Cas9, then is nickase, so that makes nick instead of double-stranded DNA break. And then base editing I talked about. And there's also CRISPR-I, which is for inhibit inactivation of gene expression. And then CRISPR-A is for activation of gene expression. And then another thing I want to talk about is these MagNucleus, so especially Mad7. Okay, and so Mad7 it belongs to Inscripta, the company, and so Mad7 is different from Cas9 in that Mad7 employs a single RNA to guide it to the target DNA sequence. And it creates double-stranded DNA breaks, however, with sticky ends. So that provides a little bit better precision in terms of gene editing, especially for gene insertion, because it's sticky ends. So only sticky ends can actually stick back together. But Cas9 actually makes double-stranded DNA break with blunt ends. So that's the major difference. And there another difference is really the PAM sequence. So Mad7 recognized a different set of PAM sequence from Cas9. So prime editing actually came out, also had a very high impact. So this is the technology belongs to now Prime Medicine. So basically, so this is a fusion. It's a fusion protein. So you can see a Cas9 protein fused to a reverse transcriptase. Okay, so this is really the enzyme. And prime editing can identify target sequence using guide RNA. Then can replace DNA nucleotide without DNA donor because the RT template can do gene replacement. So that's really the nature of prime editing is for gene insertion or deletion. So now I want to talk about TARGATT. So because I mentioned about CRISPR, all these CRISPR technology is limited because insertion in most cases is still rely on host homologous recombination mechanism. And by nature, it's very low efficiency. So now TARGATT, this gene editing platform really offers much higher insertion efficiency. So TARGATT uses integrase. It's a kind of serine recombinase, and it performs site-specific integration and enables accurate and precise insertion of transgenes in a predefined genomic locus. And so TARGATT uses two components. One is the enzyme, which I already mentioned is a serine recombinase or integrase. And the other is docking site, so which is recognized by the integrase. And this is our proprietary technology. As Lexley mentioned, I was a co-inventor of the technology when I was at Stanford University, and Applied StemCell has exclusive license from Stanford University. So the way it works is really very simple. So target enzyme or integrase recognize two non-identical sites, ATTB and ATTP. And upon recombination between these two sites, it results in integration of the transgene here in green. And at the same time, also creates two hybrid sites, ATTL and ATTR. And these two hybrid sites cannot be recognized by integrase. So that's why the reverse reaction, which is the excision reaction, does not happen. So integrase only inserts the DNA, but does not excise DNA, the insert. So the net effect is a much higher insertion efficiency. So that's really how integrase works. And so it's site-specific because it has to have landing pad recognition. And non-viral transfer, we use plasmid form and high efficiency. Another very important feature of integrase is it can insert up to 20 kb fragment. This is not really the limit of the technology. It's really the limit of how big a fragment we can actually clone into a plasmid. And publication has shown that integrase can insert large fragment like BAC DNA, which is about 100 kb in size. And then we have very clear IP and licensing terms for commercialization. Now, PASTE just recently came out, and so the company called Atome, they recently came out at the beginning of this year. That's their technology. So it's really a combination of almost all these components I just talked about. So it combines Cas9. So the enzyme itself, it's pretty complex, right? So it contains Cas9 and also contains prime editing's components, which is reverse transcriptase. And then it contains the integrase I just talked about, right, for DNA insertion. So basic components are three enzymes together. And so PASTE basically uses prime editing to insert a landing pad. I just talked about those attachments, ATTP. And then followed by insertion of DNA fragment using integrase. So basically, the system itself is pretty complex, like I said, and also requires a cell to perform these 3 steps in the right order. And you can imagine the efficiency, the end result efficiency can be compromised, right? So the cell has to do basically place the landing pad first using prime editing, and then do the gene insertion using integrase or target technology. So you can imagine that the cell has to process that in the right order. So the efficiency and also the accuracy can be compromised because the complexity of this enzyme. So I will tell you more about what we have, which is a very simple, just one enzyme that does the same thing. So first of all, I kind of like to summarize what we've talked about today so far. Is really, gene editing technology, there are pros and cons to some extent. So the current traditional way of making cell therapy products, the approved CAR T cell therapy uses lentivirus. And as you know, lentivirus inserts DNA randomly. That's a huge safety concern. Multiple copies can really cause genomic instability. And lentivirus manufacturing is not cheap and also time consuming, although it does work well, high efficiency. That's why these drugs are being used to treat patients. And transposase has a very similar problem with lentivirus, even though the random insertion part is slightly better because it seems to recognize certain sequence as better insertion efficiency. And CRISPR, of course, is site-specific. We talked about off-target, so that's still a problem that needs to be solved. And low efficiency for knock-in. So really, the CRISPR-Cas9 system cannot really use for knock-in at this moment. So if you look at all these clinical trials that are currently using CRISPR, none of them use knock-in. And so all these first drugs are knockout in nature or very small nucleotide change using CRISPR. So that's really, show that target is very, very, it's a complementary technology, right? So I'm not saying one is better than the other. It really depends on what you want to do. And if you want to insert a large cargo, especially for a car, nowadays, it's not just one car, there are multiple cars, and it easily exceeds 5 kb, for example, right? And then target will be a better way to do it because we can insert up to 20 kb and with high efficiency. And also the freedom to operate is really important when it comes to clinical application. So now I really want to share with you a very exciting next generation technology called a SELECT. And this is really something that I mentioned when I was talking about PASTE, is that basically this one enzyme that does everything that PASTE enzyme with the 3 components is doing almost, right? So SELECT is basically an upgrade of target, in which case we have evolved the integrase enzyme to identify integrase variants. And these variants can recognize human sequence. They don't just recognize ATTP site, but they actually recognize human, natural human sequence, genomic sequence. So this allows single-step insertion of large-size DNA, right, up to 20 kb. So the rest is all similar to target, but the breakthrough here is really these evolved enzymes now can recognize human sequence. And so because of that, that really pushed the boundary of gene editing technologies, and especially for integrase. And now it is capable to basically be used in all cell gene therapy modalities, including gene therapy, autologous or allogeneic cell therapy. So this further kind of give you a better overview about technology capabilities. Like I said, it really depends what you want to do. And that's when you want to select the right technologies. So we're looking at application allogeneic for knock-in therapy, I already said PACE can do that, Target and Select can do that, and not the regular CRISPR-Cas9. And then we're talking about off-target events. Both Target and PACE have better ones because we don't make double-stranded DNA breaks. There's none of the other off-target that we have observed, especially with Target. And this thing is unique to Target because knock-in for non-dividing cells. So CRISPR in general requires just the host homologous recombination system to be active. In non-dividing cells, the host homologous recombination system is not active. So that's why CRISPR cannot do that in the non-dividing cells. However, integrase is a self-autonomous enzyme and uses all, the enzyme contains all the functions. It does not require host enzyme to do anything. And simplicity. This is where I want to emphasize the simplicity. PASTE is good. It can do some other things, although it's very complex, the efficiency is compromised. And with our Select, it's much simpler and works better. Right? And large payload. So like I said, we have demonstrated we can insert up to 20 kb. None of those other technology can. So I'm going to quickly go through gene editing application. So as you know, it has very diverse application. And not only for research, for disease modeling, drug discovery, and most importantly, it is a very major application in the cell gene therapy, which is the next generation genomic medicine. So I'm gonna quickly talk about this. So application, so it's very different when you think about application for research versus clinical development. So for research, we use CRISPR, especially for knockout. So we have license from both MIT and from the ERS Genomics, the UC Berkeley branch. And it works well for knockout and small fragment knock-in. And however, for clinical use, it's pretty much prohibitory, if not impossible, to obtain freedom to operate. And so we use TARGATT to do large fragment insertion. And for clinical application, we don't use CRISPR-Cas9 at all. We actually use Mad7 to do knockout or small fragment insertion. And knockout using the Mad7 does not really, it has a very good freedom to operate because Inscripta actually has very good, they basically allow people to use their technology as long as the final product does not contain Mad7 components. So you can use the components to generate the cells. And once the cells are generated, these components no longer there, that's fine. So we use that to make knockout. Oops. And then another thing I mentioned is the AccuBase, right? So we use that for point mutation for very high safety profile feature. So we, for research, mostly focus on using iPSC. So, we streamline since we've been working with iPSC gene editing for over 15 years. So we're able to generate iPSCs, gene edited iPSCs within 6 to 8 weeks using our lines. And if you send our lines, we can finish between 2 to 3 months. And so I want to spend a couple of minutes just to talk about this. So we have created a target master cell line that can really jumpstart allogeneic cell therapy. So this is a GMP grade iPSC line. So this iPSC line basically contains target landing pad, so that's ready for repeated, fast, and efficient gene knock-in using our technology. And as you know, iPSC has the potential to differentiate and make a limited source for truly off-the-shelf allogeneic cell therapies. And and we engineered this particular GMP cell line with Mad7. to insert our landing pad, so we didn't use CRISPR. And this provide a very clear IP path to commercial. And all reagents and all the SOPs follow FDA guideline for GMP. And we have quality management systems will follow everything under quality management system. And certificate analysis and testing come with the cell line. I'm gonna skip this. That just tells, step by step, we actually how we made this GMP grade target masked iPSC using Mad7. So now we have this masked iPSC line that really contained the landing pad here, ATTB in yellow triangle here. And then starting from this line, one can insert any genes of interest in red here into the safe harbor locus and generate a large fragment insertion. So this really creates a platform which can be used for many different cell type products, right? So what we have here is a GMP target iPSC iCell platform. So basically we have iPSC, and that's with the target landing pad that can be gene edited to insert any genes of interest. And then these genetic engineered iPSC can be differentiated to many different types of cells. And these cell products can be used for cell-based therapies and can also be used for drug screening, disease modeling, and also for in vivo preclinical studies. So I'm just quickly talk about one application here, which we did, is to use our platform, the GMP grade iPSC platform, to make iNK cells. It's a CAR iNK. So started with the master cell line and then inserted a CAR using our TARGATT technology. And so efficiency without any selection or enrichment was over 40% insertion efficiency, 10 times better than CRISPR Cas9 when we did it. And also we monitored CAR expression, and it's expressed in the iPSC as well as in differentiated final products, and also all through the differentiation process. And this is just one one piece of data to show that CAR positive cells express CAR and negative cells do not express CAR. And so these CAR iNK cells can be used as a product to treat lymphoma. So this is a collaboration project with our partners. So so this is really the trend, right? So the current approved CAR T cell therapy, they are autologous. So meaning one product, one patient. They're very expensive. So $450,000 one dose. And most patients will need 2 to 3 doses. So you're talking about a couple $1 million. So what we want to really develop is allogeneic and focusing on iPSC. That gives another level of universal donor idea, right? So really provide off-the-shelf genetic modifiable cell products and provide on-demand life-saving. Talking about life-saving is because for autologous, patients have to wait for the cell to be made, and patients don't have time. So what we want to do is that these cells are in the freezer, you take out a vial, and the patients can be treated, right? So this is really, I think that's the future. I think that's everybody's dream, really. So the takeaway here is really allogeneic iPSC is a disruptive technology, although there are hurdles that we need to overcome, but we're making incremental progress towards that ultimate goal. So due to time constraint, I'm going to skip all these slides, and because we'll send these slides to you. So I hope I have given you an overview of gene editing technology, especially the most advanced technology that's available. And at ASC, we provide a comprehensive gene editing technologies with the mind of IP landscape and freedom to operate. So we use our proprietary target and select technology to do gene insertion, and we use Mad7 for knockout, small fragment insertion. We also use our AccuBase gene editing technology for point mutation or correction. And we provide end-to-end solutions. I didn't get a chance to tell you about other parts of the services. Basically provide from discovery all the way through GMP manufacturing for cell gene therapy products. And with that, I'd like to thank you very much for your attention, and I'd be very happy to answer your questions.